My name is Dr. Chris Ward from the Kansas University Medical Center. Today I want to talk about how to transfect plasmids into the human 293T cell line using a cationic polymer called polyethylenimine, or PEI for short. So why would we want to do this? Well, we want to transiently transfect a plasmid into a human or eukaryotic cell so that the resultant protein is properly folded and glycosylated and is similar to the native protein that you'd find in your body. Now we want to do this because we might want to look at interactions between two different proteins, or we might want to just make a lot of a particular protein so we can screen monoclonal antibodies on it. So the idea is to insert a plasmid into the cell, and this plasmid has a very powerful promoter on it that will run off your favorite gene and make quite a bit of it in the cells, and then you can isolate the protein just in a simple lysate of the cells, or you can make a membrane prep. So the first thing is, where do we get our polymer? So we get our PI from Polysciences, and there's the catalog number. We dissolve the powder in water and then adjust to pH 7 with hydrochloric acid. So initially the solution is cloudy, but as the pH is adjusted it becomes clear, and this might take some time to process. It can take several hours for the PEI to go into solution. We then adjust the volume to a final concentration of 1 megamil, and then we filter through a 0.22 micron filter to ensure sterility and to remove large pieces of insoluble PEI that might sequester the DNA. We make 1 mil aliquots and store it at -80. For every batch of PEI, we test the transfection efficiency using different DNA PI ratios, 1:1, 1:2, 1:3. I usually use a GFP expressing plasmid. So this makes a nice green fluorescent protein that you can look at using a fluorescence microscope. And that will give you the approximate percentage of the cells that are making that have been transfected and are making protein. And I can get up to about 80%. So what tissue culture reagents do you need? Well, you need RPMI with L-glutamine as a sort of basic feedstock. To starve the cells, we use OptiMEM, HEPase, and sodium bicarbonate in L-glutamine. We also need fetal bovine serum, pen and strep, and Glutamax. The pen and strep is basically to suppress any bacteria that might be there. The DNA is not sterile. So here's the protocol. We plate the 293T cells in 14 centimeter plates in RPMI 1640, 5% bovine serum, penicillin and streptomycin and Glutamax. So we usually use bovine serum, but you can use fetal calf serum. It's a bit more expensive. When the cells are 80% confident, remove the media and replace with 12 mils of OptiMEM and incubate for 2 to 4 hours. This kind of starves the cells and makes them more pinocytotic so they can take up the complex. Then in a separate Eppendorf tube, we add 16 micrograms of plasmid DNA to 0.75 mils of OptiMEM and 0.25 mils of megamil PEI. We then incubate the complex for 15 minutes, and the complex we should get sort of cloudy suspension as the complex is formed. And then we add the complex to the medium to the plate in a dropwise fashion. Then incubate overnight, and then add 12 mils of RPMI 1640, 5% bovine serum, penicillin, streptomycin, and Glutamax. And then you can incubate 2 to 7 days. And during this time, your cells will make a lot of the protein. Now, if it's a secreted protein, you want to go for the supernatant. And if it is one that is a membrane protein or intracellular, then you'll need to go and isolate the total cells, either make a total cellular lysate or a membrane prep. And so these are the two papers that describe the protocol. One is Raymond et al. And the other one is Christina Ehart et al. And both go into a bit more detail about the protocol. But this is a bare bones protocol that we use day in and day out. So now we'll go into the lab and we'll actually do the protocol and see how we get on. We plan to take that plasmid that we're using, the AH8 flag, and we're going to transfect some 293 T cells, pink cells. Now these are about 80% confluent. As you can see, they've used up much of their medium. What we're going to do is we're going to take the medium off them and starve them for about 2 to 4 hours. Probably 4 hours is better. And then we're going to make a cocktail of our DNA with this stuff here, which is polyethylenimine, which is a long lipophilic cationic polymer, basically. And so imine groups will bond with the phosphate of the DNA and kind of encapsulate the DNA in a hydrophobic coat, basically. And then this will then interact with the cells and introduce the DNA into the cells and then ultimately into the nucleus, where it will be transcribed, at least transiently. So the idea is to take 4 large 14 centimeter plates and transfect them all, because we need a lot of protein, because we're going to use it as a Western target for screening monoclonals. So we've vaccinated some mice against the plaque domain of polycystin 1. And this DNA will contain the plaque domain, and when we express it in the cells, we'll get a big protein of about 110 that's got a flag tag at one end, and got the rest of C-terminus of polycystin basically at the other. And it contains the plaque domain to which we've raised the antibodies. So this is just making a properly mammalian expressed protein in a sort of an appropriate cell line, and then just making a crude membrane prep that we'll then use to screen our antibodies. So the first thing we've got to do is just take the medium off and then replace the medium with OptiMEM, which is really designed for things like Fugene. It's optimized for transfections. And so we'll take the medium off and we'll just replace with 12 mils of OptiMEM. And then we'll starve for 2 to 4 hours, and then put a cocktail of polyethylenimine and DNA on top of them and leave that overnight. Okay. So, just take the first, right, put it open like this. Try to keep it bug free as much as possible. Of course the DNA is not going to be sterile, but usually we get away with it, at least for 12 hours. So we'll do the transfection overnight, and then we'll actually feed them with antibiotic containing media, keep them going for another 3 days, maybe 4 days, so we get a maximum amount of protein. Because of course we want this as a target for our monoclonal screen. I don't know if we're going to be successful in this, but it'd be nice to have a nice monoclonal to the plaque domain. We might be able to use, for example, a hyper resolution to see where 7012, for example, and the plaque domain antibody actually interact with reference to other proteins, for example. So we need this plaque domain antibody. So I'm just going to replace the medium here with the OptiMEM, just 12 mils of it. Okay. And then gently put it on top. Don't want to disrupt the cells too much. Just push it on the side. Don't touch anything, you don't want to plug anything. don't know what starving does to the cells. So some people say it makes them more anisotropic. I believe that, and that helps take up the DNA pi complexes. I'm not sure. I just know that it does help. So hopefully we'll get a really good signal from this, and we can use it to screen our monoclonals. So we'll probably have 1,000 monoclonals to look at, or 1,000 wells, probably about 400 monoclonals to look at. So it's quite a bit of work. But you never know what you're going to get. You might get the next 7012, or you might get something completely non-specific. But that's the fun of it. Okay, and I'm just going to put these in the incubator. Oh, and yeah, one thing for the students, when you're actually putting something in the incubator, just kind of pop it in, make sure it's nice and even there. These are kind of even. And then just make sure there's some water down here. This is copper lined, it kills bugs. And then just remember to seal it like that. Okay, that's really important. Some people just leave it open. The carbon dioxide will just fall out. You just want to shut it and close it. And then just watch your carbon dioxide come back up. It should go from 3% up to 5%. And the temperature should come back up again. And then we'll see it in 4 hours. Okay. Hi. So now we're going to make a mixture of DNA. So we've got the flag AH8, okay, at 0.7 mg per mil, right? So we need about 16 micrograms per plate. So we're going to use about 23 microliters of DNA, and we're going to mix that with 750 microliters of optiamium. So I always like to aliquot what we're going to use first, so I don't contaminate the bottle. Kind of expensive stuff, this. Just pin those or so. And reach it. Now, each tube is going to get 750 of optiamium. So this is what we put on the cells. It's just optiamium with no antibiotics and no serum, no additives whatsoever. Basically starvation media. And we take our DNA and we just want 23 micrograms in each. Filter tips. That's 23. One. Two. Three. And 4. Quick mix. So this is the C-terminus polyethylenimine with a flag tag on the end, and of course a signal peptide to get it into the ER. So each of these gets 250 microliters of pi, and I'll explain that later on how it's made up. So polyethylenimines, 250. So that's enough for one of those big plates. Each tube is enough for one of those big plates. So, again, try to get a quick mix actually. Big fog. Straight in. work okay. Maybe not optimal, but yeah. So it starts off clear. But with time, it should kind of go cloudy. I do it 15 minutes now, just at room temperature. And we should be fine. Okay, so we're going to transfect these cells with AHA overnight, and then we're going to feed them with antibiotics containing medium and fetal calf serum tomorrow after about 18 hours. So all we have to do is take the starved cells, like these, and take a tube of this, and you can see it's kind of cloudy-ish. Don't know if you can see that. There we go. It's kind of cloudy. That's what you want. It just means that the DNA is kind of interacting with the polyethylenimine and making a complex, and then we can, and that complex should transfect the cells. We'll do this, and we'll see how we get on. So this is a very crude transient transfection. We just want to get the C terminus of polycystin expressed in these cells at high level, so we can use them as a target for monoclonal screening. We just do that. And mix. We just sprinkle it around. Just be kind of gentle, because these 293T cells will come off really easily. Now, interestingly, after you've transfected them, they kind of stick like they seem to stick a bit harder than usual. You've got to be aware of that when you're harvesting. We're going to give them 3 days to make this protein. And, I happen to know it expresses quite well, and we should be able to use it as a target. So this is human polycystin 1, and we're raising mouse monoclonals against a human protein. Now that's important because there's 98 million years between us and the mice. Now if you try to make it in a rat versus a mouse, there's only about 20 million years. Same for guinea pigs and hamsters. So you want the biggest phylogenetic difference you can. And also, this is a human disease, it's not a mouse disease. And we're going to use these antibodies to look at human cell lines and human urine. You know, and the mouse is a deprecated organism for studying cystic disease. There's a lot of reasons for that. If you're a heterozygote PKD1 mouse, you're completely normal. Whereas if you're a human being that's heterozygote, you're in deep trouble. And you're going to go into renal failure in your 50th decade. And furthermore, with PKHD1, which is a very severe polycystic kidney disease, the recessive form, the mouse doesn't get a renal phenotype really, very mild cystogenesis. But if you completely knocked out the PKHD1 gene in a human being, that baby, I think there's only been a few case reports of people that are completely null for PKHD1 surviving because they have massive renal cystogenesis, which you just don't see in the mouse. So I work on humans, human urine and human cells, and I only use a mouse to check things, to check if a gene can give rise to a cystic phenotype, for example, or to make monoclonals in them. I'm certainly not going to try to do any serious biology in them. We'll do that with cell lines and by studying individuals that have got the disease. That's my take on it. So again, I'm just going to shoot these very gently again. Pop in. Good. Right, there you go.